excited about what we're going to be talking about. And so I wanted to tell you right out of the gate a little bit about myself. So I'm a longtime teacher, originally from California in the Bay Area where Bio-Rad is located. But most recently, I've moved to the green state of Vermont, and so, over on the other side of the country. And so I'm kind of introducing Vermont to biotechnology, and it's been kind of fun. And so I am going to give you a little background on one of Bio-Rad's most interesting kit, I'm going to say, or series of kits having to do with cloning and sequencing. And then I'm going to talk a little bit about the details of that, the student lab protocol, and then sort of culminate with some projects that I have done with students with a variety of Bio-Rad kits. And so it's an opportunity for when I'm going over something, you can think, is this something that would be really cool for my students to do? And like Tamika said, if you have any questions, you can put them in the chat or even just shout out, and I'll try to answer them as we go. So here we go. Let's check this out. So the cloning and sequencing series of Bio-Rad, it's a 6 to 8 week series of activities made up of 8 different modules. And so it takes quite a while, and it's a totally comprehensive and interesting workflow that really gets the students to engage. And there's lots of reagents, enough for 12 workstations. And then it culminates in the sequencing of a gene. And so you would then take your DNA samples and send it out to a sequencing service. And I'll talk a little bit more about that as the slides go. And so why teach cloning and sequencing and engage the students this way? Well, there's a variety of reasons to do that. One of them is that when students conduct novel research and they're on the edge of discovery, it's most intriguing. Just like the reason why science is an exciting profession, it's because the outcome are unknown, and so the students are discovering, they're making decisions, they're analyzing their data as they go, and they're formulating explanations based on the data that they're collecting, and they can sort of pivot one way or the other. And I'll try to give examples of that as we go through. It's hard to just generally speaking about that. These experiments are not simulated, they're real. And so it's challenging, and sometimes the results aren't working, but as you may know, when something isn't working, it reveals something about what could be done differently, et cetera. And so there's some totally important information that's gained. And so the culmination, just to sort of get to the chase of it, the culmination is if the students extract DNA from a plant that they're studying and amplify it, and ultimately they want to sequence after cloning that gene, and then their sequencing data can be uploaded to the National Institute of Health, or NHI, the National Center of Biotechnology Information, or NCBI. And so the students can publish their data, and depending on what organism they choose, it may be completely novel. And so they're adding, just as an authentic scientist would be adding their data to a collective in the National Gen Bank. And so it's a real accomplishment. And so I hope that's intriguing to you. And so just a quick overview of what we're going to be talking about, and I'll go into detail in each of these as time goes by, and feel free to ask questions again. But basically, it begins, as you can see over here, with an extraction of DNA from a plant. And then the DNA a particular gene is amplified using PCR, and then it's purified. And the gene, spoiler alert, is glyceraldehyde phosphate dehydrogenase, and more about that later. So the gene is amplified, and then those amplicons, those copies of the gene, are then sutured or ligated into a plasmid vector, which will then be used to transform bacteria or E. coli. And when the bacteria grow in an outculture and the numbers increase, basically they're cloning the plasmid. And so you're getting many, many copies of the plasmid. And then it culminates in what's called a mini prep, where the cells are lysed and the plasmids are then released and purified. And then in order to verify whether or not the insert was properly ligated, a restriction enzyme digest is conducted. And then if that is successful, then you would then add sequencing primers to your plasmid and then send it in for sequencing. And then you'd get your sequences back, and then you'd analyze it using bioinformatics, and you'd put together various contigs into one continuous sequence. And so it's quite an endeavor, and it's totally awesome. And so I wanted to point out that if you have background in it, great. And if you're just intrigued by it and you want to plunge into it, Bio-Rad has the most comprehensive and thorough manual, bar none. It's over 350 pages, the PDF, and there's a link to it in the slideshow here. But it goes through each of the modules, and it gives detailed information, not only for the instructor to figure out what's going on, but the resources that are provided in the kit, the reagents, what equipment that you have, some background information, all kinds of stuff that you would need. But the first module is a DNA extraction. And the second module that comes out is two types of PCR. So it's a PCR and a PCR clean, and a more detail about that. And then the third kit is gel electrophoresis, agarose gel electrophoresis materials. In other words, buffer and sugars, etc. There's 2 times in the workflow that students will need to conduct agarose gel electrophoresis to verify that the results are progressing as expected or predicted. And then there's a PCR product clean kit, and for which more about that in the future. But when the amplicons are created, they're then cleaned before ligation. And then, speaking of ligation, there's a ligation kit that comes with containing enzymes to ligate into the vector, and there's also transformation involved. And so there's nutrient agar, there's ampicillin, there's plates, there's inoculating loops in order to grow bacteria and transform them. And then there's a plasmid, a mini prep plasmids, where the students will then outgrow the cultures into larger numbers so that they express more plasmid, and then the cells are lysed. And then the plasmids are purified using columns. And then the final module is a sequencing module where they'll add primers to the plasmid and then send it out for sequencing. And so I want to hang out with this slide because this particular slide really gets to the heart of all of the techniques that the students will be conducting. It's quite comprehensive. And that's why at the beginning of the presentation, I mentioned that it takes several weeks. This particular workflow could be, depending on what you're thinking when you're hearing this, if you're hearing it for the first time, it's certainly, it could be a semester course or an entire course, depending on how you want to approach it. I can tell you a little bit about, at the end, how I approached this with my students. And I did create an entire course around this particular series. And so the techniques that the students will acquire will, of course, be the techniques of DNA extraction, which is very important in all realms of biotechnology, where DNA is precipitated out using ion exchange chromatography, and I'll get into that a little bit in alcohol wash. And then quantification, in other words, determining the purity of the DNA. That's a technique using a spectrophotometer or a nanodrop. The students will acquire the skills of PCR. They'll do 2 rounds of PCR, a broad PCR and then a nested PCR. And then they'll chop up extra primers with exonucleases. And then to make sure that the products were amplified properly, students will conduct agarose gel electrophoresis. They'll learn how to do that, if not already. And they'll look at band identification in order to identify whether or not the PCR was successful. And then they'll do PCR purification so that the ligation occurs properly. In other words, they'll clean using size exclusion chromatography, and they'll remove unincorporated DNTPs and primers in order to get the pure amplicon. And then they'll conduct a blunted ligation. They'll be adding a special type of polymerase that removes an adenine hang on their amplicon. More about that later. And then they'll use the enzyme ligase to ligate it into a circular a piece of DNA referred to as a plasmid. And then the plasmid will then be used to transform E. coli. And then those cultures, if they're grown, they'll be grown on ampicillin plates. In other words, there's an antibiotic selection, and there's also a positive cloning selection. I'll talk about that. And so the students will learn about microbial cloning and culturing, as well as mini prepping, which is a small version. You could, if you're working with large amounts of cells, there's even things called mega prep and giga prep, but this is a mini prep. And then those cells will then be lysed and then restriction digest. And so they'll learn about how restriction enzymes can cut pieces of DNA. And then another gel is run to verify that. And then sequencing primers are added to the plasmid, and then it's sent in for sequencing. And then finally, the sequences get returned to you. And then students will analyze those sequences in the process of bioinformatics. So there's a lot going on. And so feel free to ask questions and interrupt if necessary. So here's some background information. So why plants? Well, why not plants? They're great to work with, and there's a number of species that are available, and you can use traditional crops. Students can choose plants from around their house to study. You can go out into nature and look around the school campus. And so there's a lot of diversity, and depending on your specialization or expertise or your love, you can look at those sequences when you gather them and sort of take a phylogenetic approach and try to analyze. There's a lot going on here. They're just easy to work with. There's not a lot of ethical considerations when you're talking with plants, and there's no approval needed. And so what gene are we talking about? I mentioned it before, but you want to begin a project like this by first looking at a housekeeping gene. And what that means is a gene that is highly conserved in nature. And so most organisms, no matter what you're talking about, has it, and it's always expressed, and that it codes for a protein that's really critical. And so some examples of that are glyceraldehyde phosphate dehydrogenase, GAPDH, which is the one that we're going to be working with. But other examples of a housekeeping gene would be ATPase. It's the enzyme that converts ATP to ADP. Most organisms have that, and there's genes for that. Cytochrome C, again, a great example of a housekeeping gene. Most organisms have the electron transport chain, and that's that gene is producing a cytochrome C involved in that. Beta-actin is also a very common gene, and it produces the actin protein, which is common in microfilaments. It's important in muscular contraction and cytokinesis, and go on and on that. So those are common. But we're going to be working with GAPDH. And so GAPDH, if you're not familiar with it, it is it's involved in glycolysis as well as a number of other things. And so as you can see here in the slide, it's involved in DNA replication. It's involved in membrane fusion, apoptosis, which is programmed cell death. It's even thought to play a role in Alzheimer's and cancer. But it's probably its highlight is that it is involved in glycolysis. And this is a simple diagram of anaerobic respiration. You can see that glycolysis is 10 steps leading to pyruvic acid. And then without oxygen present, there's a redox reaction that either produces ethanol or lactic acid. But if you look at glycolysis in detail, in the 6th step, you're looking at glyceraldehyde phosphate is the substrate, but the enzyme GAPDH is a triose phosphate dehydrogenase, which means that it's catalyzing this redox reaction of this sugar, and it's also phosphorylating a phosphate group because of the redox reaction. So it's a critical enzyme in glycolysis. It's one of the most common pathways in nature, and that enzyme is right up in front getting it done. And so that gene, GAPDH gene, is a great one to look at, because you know it's going to be present in all different types of plant species that the students want to study. And so what's interesting is, what is the gene sequence? Are they exactly the same? Unlikely. Even though the proteins may be conserved, you may know that they're over long periods of evolutionary time, there's slight changes that can occur in the genome, as well as the fact that the gene, we're looking at genomic GAPDH that produces the protein found in the cytoplasm, but there's some interesting things. There's some enzymes found in chloroplasts as well. And so there's a lot to consider. So let's look at the student lab protocol and see what gives here. So we're going to first take a look, a more detailed look at the amplification of, or the extraction and amplification of the gene. And so DNA extraction, boy, is this important. And so there's many levels of DNA extraction. You can do it very simple using just simple like salt and dish detergent and crushing, but this is a real advanced one. And so basically what you're looking at here is you're going to be grinding up plant tissue to sort of break through the cell wall, and you're going to be using spin columns and a centrifuge by using ion exchange chromatography, and then you're going to then elute the DNA using alcohol. And so the detail of this is that cell walls need to be crushed. And so you could use a porcelain mortar and pestle if you're using large samples, but the kit provides sort of this, what you'd call a micro pestle, which is basically a microcentrifuge tube in this plastic pestle here. And so you have to get physical with the cell wall. And so you have to crush it. But then the lysis buffer has a potpourri of different chemicals. And I find myself, and this slide's a bit busy, but I find myself being very interested in the chemistry of the lysis buffer for whatever reason. I think it's really cool, and the students get into it as well. And this is an opportunity where you can sort of really slow it down and really get reinforce all kinds of concepts with the student. And so one of the things that the lysis buffer, which means the chemicals necessary to break the plant cells open, it has this chemical called EDTA, and it's a chelating agent, which is really important at binding cations, especially magnesium, that nucleases, when the cells are broken open, it sort of ruptures the cell, and vacuoles containing nucleases or enzymes that will break down DNA get released. And so this particular chemical causes them not to be able to work because they need magnesium in order to function. And so the lysis buffer also contains a buffer that is often called tris. Tris maintains pH because some of those vacuoles release acid into the environment, which is unwelcome. And then there's a variety, depends on the lysis buffer, but you could use any kind of detergent or CTAB, which will then disrupt the cell membrane and also the nuclear membrane of the cells in order to release the nucleic acids or DNA. And then there's DDT, which is added toward the end, which is a reducing agent, which minimizes protein oxidation. Little bit later in the presentation, spoiler alert, I had students working with redwood trees when I was in California, and there's some issues with redwood needles. And so we had to really break apart this lysis buffer and look into the literature and consider what other lysis buffers contained. And it was an opportunity. And these are things that I was learning as we were going, and the students were really like intrigued by that. They're like, well, you don't know this? And I'm like, no, I don't, but we can contact different universities and we can look at and talk to those researchers that are working specifically on those particular plants. And the students were just like, they just fell in love with the fact that things were unknown and that they needed to talk to college professors to get answers. It was really cool. And so I'm going to be brief with this, that once the cells are lysed in the lysis buffer, then that the lysate is then applied to a column, which is, I mentioned, is ion exchange. And there's beads in here. And the theory behind this is that as a result of the DNA being a polyanion, and it forms a cation bridge depending on the salt concentrations. And so DNA will attach to the beads, and then other unwanted material and debris and protein are washed from the column, and then alcohol is added in order for the DNA to precipitate out in solution. And then when the DNA is released from solution, in order to think, well, do I have it or don't I have it? Is this good quality DNA? Then you can use a ultraviolet spectrophotometer at a 260 nanometer frequency to determine the proper absorption of pure DNA. Or if you have access to a nanodrop, you could use that as well in order to determine the concentration of DNA to see if it's pure or not. This is also an opportunity. You might say to yourself, well, I'm not sure if I have this level of equipment. It's an opportunity for you to then send your DNA samples out to a local university or research center close by and start to build a collaboration. Another cool opportunity. So then, okay, you successfully have DNA from a plant. And so now you want to use PCR to amplify the gene. And so you're like, well, how can we do PCR if we don't know? Well, we need primers, but if we don't know what the sequence is of GAPDH, how can we do PCR? Because normally you would use very specific primers that would anneal forward and reverse to the target sequence that you're trying to amplify. We don't know what the sequence is of GAPDH if we're using whatever plant. So degenerative primers are used. So a series of about 12 or so primers are in a mix that are used. And you're like, well, how did we come up with those primers? Well, because of homology of between plant species, there's a similarity, and we do know some sequences of GAPDH. And so these primers, that's a short story. These primers are added, and they're able to amplify the gene, even though it's a plant that we don't even know what the sequence is. But as a result, the amplified target sequence may not be robust. And so after one full round of PCR, Then the amplicons are treated with an endonuclease to get rid of those primers. And then another set of primers are added to the PCR product. So now you're working with an amplification of an amplicon. And so it's very robust. And so that nested PCR, as it's referred to, is very useful when you're trying to analyze whether or not you're successful using gel electrophoresis. And so a little sidebar on PCR, because it's really important. PCR, polymerase chain reaction, is basically DNA replication in vitro in a tube. It's very specific, and it can make millions of copies of your DNA, and that's what we're trying to do. We're trying to take template DNA from a plant and add primers that will be forward and reverse, and using a polymerase enzyme, and it needs to be heat stable, because in order to open DNA up for replication, you're going to use tremendous heat, like around 95 degrees. And so scientists have been using TAC polymerase, which stands for Thermoaquaticus, which was isolated from a bacteria in Yellowstone National Park, so it's heat tolerant. And then DNTPs are added, of course, in order to make more DNA. And then there's a cofactor that needs to be added, magnesium chloride, and some buffers, because of the phosphorylation that occurs in the pyrophosphates during replication. Things get really acidic, and so the buffers kind of keep things under control. And so PCR really quickly is taking your sample and heating it so that the DNA opens. The hydrogen bonds between the nitrogen bases will just like basically open up like a book. And so it's really hot, 94 degrees, the DNA separates. But then it gets a little bit cooler, not exactly cold, 60 degrees, and that allows for the primers to attach or anneal, therefore bookmarking the region that you're trying to amplify. And then the third temperature, there's 3 step temperatures here. The third one is a little bit warmer, 72 degrees, and that's the optimal temperature for TAC polymerase, which is the enzyme that replicates DNA, to then start extending. And so what happens is when the primers attach, the polymerase starts extending, and then after 2 cycles, you get 2 copies of DNA and then 4 copies of DNA, and it just keeps replicating like a chain reaction, and you get many, 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 many copies. And so millions and potentially billions, depending on how many cycles. And a cycle is made up of the hot, the cool, and the warm. And so as you can see here, if you run it, this is the cycling parameter. If you do it 40 times, the first temperature for 2 minutes is just to sort of get it to open. And then 40 cycles, that's a lot of DNA. And then it finishes with a 10-minute, like, okay, let's just roll with extension and see how much we can make. And then there's an infinity cool setting for when you're done. So if you're successful, you will have amplified the GAPDH gene. And again, I mentioned this before, you're using degenerative primers that account for the variation in DNA sequences between whatever plant that you decide. And then you'd be using a nested PCR. So the template for nested PCR is the amplicons from the first PCR. And so that's pretty cool. And so once that takes place, and here's a diagram of that, the first primers are sort of outlining the target area and making the first amplicon. And then the second amplicon has a different set of primers, which are going to be a little bit more specific to the region that you're interested in. And so it improves the specificity and the sensitivity, and it blocks out any sort of non-specific amplicons that are in the mix. And so I like this picture right here. It shows that initially you're looking at this, but then in nested PCR, you've got many more targets. And so therefore the results are way more intense and bold when you're running an agarose gel. And here's an example of that when you're running an agarose gel. you can see here the banding patterns right here. And so basically, without getting into sizes of the amplicons or anything, at this point in the investigation, the students are just kind of, and you are thrilled with the fact that, okay, we've extracted DNA and we verified that the concentration of the DNA is strong. And then have we successfully amplified the gene? And when you run a gel using agarose gel electrophoresis, this banding pattern right here indicates that you were successful. You can see how the nested PCR, do you notice how much more illumination occurs because there's more product. And so things are looking good. So after PCR, you want to then clean your product before ligation, which is inserting your amplicons into a plasmid vector. So PCR purification, very, very important. It's using size and exclusion chromatography, and the story with that is that these beads in the column are kind of like wiffle balls, if you can picture that, a plastic ball with little holes. So anything small is going to get caught up in those holes and move slowly. Anything large is going to sort of worm itself through the beads and elute faster. So as it says here, small molecules like the primers or even primer dimers, which is where primers anneal to themselves, which you don't want. All your unincorporated DNTPs and enzymes get trapped in the beads, but your product just flows right through, and then you capture it. And so that's a PCR clean. Now that you have clean amplicons and you're ready to go, and you're excited because now you're going to try to put it into a vector. You're going to try to ligate it. You're going to ligate your GAPDH amplicon into a plasmid. And then once you've done that, you're going to transform it into bacteria and then grow it up. So what's interesting about this is the type of ligation that the kit calls for is a blunted ligation, which means that there's no sticky ends. Sometimes when you're putting an amplicon, I apologize for the complexity of this, but if you're putting a segment of DNA into a plasmid, sometimes a restriction enzyme is necessary to cut the plasmid, which creates a sticky end. And then the amplicon is treated with the same enzyme and it ligates in. But this time, a blunted ligation, and I'll explain more detail when we get to that. But the blunt is prevented because the Taq polymerase enzyme that we've been working with is from bacteria, and the truth be told, though it's heat stable, it's not a great proofreading enzyme, and it makes mistakes, and sometimes what it'll do, it'll leave an adenine on the 3'. It's a little detailed, but it'll leave a little overhang on the amplicon. And so there's a one little skip step before ligation that the students need to conduct, which is adding a different proofreading polymerase, just a small amount to that, and it'll proofread or cut off the hanging A to it, sort of like a little hanging chad. And then the enzyme ligase, which was acquired from a bacteriophage, that enzyme sutures the amplicons into a circular piece of DNA, which is already linearized. And so it catalyzes a phosphodiester bond. And so this particular plasmid vector is called P for plasmid jet 1.2. And so Bio-Rad has chosen this particular plasmid for a variety of reasons. It's been designed for cloning, and in particular, it's designed for blunt end cloning, and it's already linearized. And I know that this is a lot to take in, but if you can see here in this green area here, so it's not closed, so it's already open, but it's sort of drawn as a circle, but it's a linear piece. This green area right here is referred to as a multiple cloning site, which means that it contains, by design, many restriction sites so that restriction enzymes are capable of cutting the plasmid, which is going to be important. I'll come back to that. There's also a gene over here on the left side, as you can see here. It stands for beta lactamase, or just simply ampicillin resistance. We want the bacteria E. coli, when we transform them by putting the plasmid into the E. coli, we want it to grow on an ampicillin plate in order to make sure that our colonies are free from contamination. And so that's important. And then this purple gene, really cool. It's called ECO471R gene. It's a gene that if the amplicon, their PCR product, does not ligate or insert into the plasmid, the plasmid would close in on itself. It would self-ligate, and if that purple front of the gene connects with the backside of the gene, it'll actually produce a protein that will kill the cells. So if we're successful and we ligate the gene into that plasmid, the cells won't die. And so it's a positive form of selection. Hopefully that was clear. So this is a good diagram of that. Here's our PCR product. What we want is to successfully ligate or insert the fragment into the plasmid. And if it does get in there, that means that the cells that are transformed will not die. And if it doesn't get in there, the cells will die. And so if there's no growth, there is no transformation. And so there's some couple of things that can happen here. And so this is, again, at the front of the presentation, I said that it's real life science. Students are like, they're in it. They're in the kitchen. They're they know what's going to go on. And it's like they need to try to analyze what could happen, what might happen, what is happening. And so there's different things, there's different possibilities. I mentioned this, that if the plasmid itself self-ligates, then the bacteria are going to die. That's one thing. Sometimes the PCR products will ligate to themselves, which is not welcome. You want them to go into the plasmid. Sometimes primer dimer will self-ligate, and you don't want that because you have the enzyme ligase in there. And sometimes there's multiple copies of your amplicon that will occur adjacent, and you don't necessarily want that, multiple inserts. What you want is just a clean vector and insert. And so when you have that, you then transform E. coli. And so you make the cells competent. If you're familiar with the transformation, this module alone is something of very importance in biotechnology, transformation of E. coli. And so you're putting plasmid into competent E. coli cells using calcium chloride to make them competent. You're chilling them. You're then using a heat shock technique where the plasmid then goes in, and then you streak those on a petri dish with ampicillin plate, and then you grow them. And if they're growing, you're like, these are our guys. These are our E. coli. They have been transformed. They're growing on ampicillin. That's one indication. That's good. And the fact that the bacteria are alive must mean that the plasmid took the insert perfectly and the cell and ligation was successful. So when you grow cells on a petri dish, I know there's millions, but these colonies could use a little outgrow, if you will. And so this cells are then transferred into culture tubes, and they're grown in a shaking water bath at 37 degrees, and the cells increase and increase and increase. And then what happens is you'll want to then lyse the cells using a mini prep, a spin column. And when the plasmid is then purified, when the bacteria then break open, a restriction enzyme is added to the plasmid to cut it at those restriction sites that I was mentioning before. And then a gel is run to verify whether or not the plasmid is exactly the way you want it to be. And so here's a little bit more detail in that. And so once you have successful colonies, and these are all like really cool eureka moments. The students are like, it's motivating them as they go. Once you have that, then you'll culture the cells, and you'll grow them up, and then using a mini prep, you'll then purify and lyse the cells. You'll get the plasmid, and then you'll apply a restriction enzyme digest to the plasmid and cut it. And when you cut the plasmid, you'll then run a gel to make sure that it's being cut. And so this diagram helps this conversation. So here's the plasmid, here's the insert, and there's a restriction site BGL2. When you apply that restriction enzyme, it'll cut right there and it'll cut right there. So what it will do is it'll release the gap DH into a small piece. Here it is right there. So it's a small piece, and then the rest of the plasmid is much larger. So when you run a gel, what you see is here's the small piece, and so that's good. And then here's the larger piece of the plasmid. So if you have two lines, that means things are really looking good. And so the students are using gel electrophoresis in order to verify a successful outgrowth and mini prep. Here's some pictures of the actual gel electrophoresis from above. And so you can see the two lines here. You can see how lane 2 is the mini prep digested with the restriction enzyme. And then over here in lane 3, I'll just look at 2 lanes. Lane 3 is the plasmid, but it's undigested. So there is no restriction enzyme. So that's the whole plasmid. So that gives you the size of the plasmid. And then when it's cut into 2 pieces. And this is using a 1% gel. So once the plasmid is good, now of course I didn't mention this, but it would be silly if you added the restriction enzyme to all of your plasmids. You would cut your plasmids and your whole experiment would be destroyed. But you're only using a little bit of your plasmid from the mini prep just to verify whether or not it's good or not. But all of your other plasmids, here, let me go back a little bit here if I can. All your other ones, in other words, all of the good ones like this, are going to be now sent in for sequencing. And so the final module is that you add sequencing primers. And so these are going to be 2 forwards and 2 reverses. It's a large segment, and so it needs to amplify part of the vector plasmid and also the amplicon. And then those are then pipetted into plates, and then those are sent into a sequencing center using Sanger sequencing, which is don't want to go too far with that, but it's using dye terminator sequencing. It's a little sequencing. Sanger sequencing is a little bit similar to PCR, but it's using a dye that sort of interrupts the PCR, and it creates multiple fragments, and then run through a gel. You can determine, or a fragment analyzer, you can determine the sequence. That's a short story on that. But here's a picture of the primers in the sequencing series. You have 2 forwards. You have one for the PJET sequence forward over here, and then one for the gap sequence, and then you have a gap reverse and a PJET sequence. And so those are the 4 sequencing primers. And then they get sent in. There's, the sequencing center could be any sequencing center. It could even be a local university that would run your samples for free if you have a connection there. Or it could be there's one called Genius, and there's a free trial if you sign up for that. But if you're really getting into it and you really want you want to expand this dimension with your students in bioinformatics and analysis of sequence, you might want to get a subscription to this site. And but basically, the data gets sent back to you in a file, and you open it up and you basically see the DNA sequencing. If you've never seen a chromatogram before, it's pretty awesome. It looks like this, and you can see the A, T, C's, and G's right here. This analysis of the chromatogram and bioinformatic would needs to be an entire webinar in and of itself. But basically, the software will cut out and trim areas in the front and in the back that don't sequence well. It'll analyze and remove introns, and then bring exons together in one continuous sequence, and so that you have a sample. I can actually show you, before I get into student projects, I think I actually have something over here. Bio-Rad has on its website, it has some sample of broccoli. And so this is what it looks like when the samples are sent in. It's really cool. And then you can sort of toggle between forward and reverse sequences. And so when I was getting into this with my students, it was I'll never forget it when I got the email that the sequences were done, and they were successful. And I was like, I almost fell off the chair, and the students were like, literally could see it in my face as they were coming into class, and they were all hugging each other, and they were so invested in this project because they'd been working on it for such a long time. And so we could pause now if there's any questions on this, or if Tamika, if there's any questions in the chat. What I want to sort of segue to the final part of the presentation is just to show you a project that I ran with my students that I was mentioning with Redwoods, using the cloning and sequencing series. And then I wanted to finish by bringing to your attention that it doesn't have to be particularly cloning and sequencing. It could be any of these biorad kits can be modified and tweaked and angled into independent research that students conduct. And it's independent research that's made easier by the fact that all of the reagents and protocols are in place. But it doesn't mean that the answers are there. It just means that the protocols and reagents are there, and the students have their own questions, and they discover and they explore. And you know, to be corny, but corny alert, that's what this is. This is Bio-Rad's Explorer program, and it really is. So if I can take silence as no questions, or I can take some questions.
Feel free to unmute if you just like to verbalize your questions, if you have any at this point.
Nope, I'm good. I don't know about the rest of you.
Everyone's hanging tough. Well, okay. I'll just say
I do
that,
have a question. I'm sorry.
okay, go ahead.
What type of expenditure did your school have to put out in order, not just the kits, but the other equipment?
Yeah.
The centrifuge,
Right. That's an excellent question. And so there's assumptions here about, you know, that your lab or your school has some basic things, like it'll have a thermal cycler, and it's like, well, we don't have a thermal cycler. And it's like, well, you could take samples or you could there's a loaning program, and certain districts have that they share equipment. I know when I was beginning my process, I didn't have a good enough spectrophotometer to analyze the DNA, and so I called up Bio-Rad, and they sent one in the mail, and I used it, and then I sent it back. And then I said, well, wait a minute. And I might as well explain this. And when we start applying it to redwood trees and multiple trees, you start needing to use a lot more reagents and the cost gets really high. And so living in Northern California, I discovered there's an organization called Save the Redwood Trees. And so I set up a meeting and I talked to them and I wrote a grant proposal, and they awarded the students and I, over the course of a few years, several thousands of dollars, which really helped. But things like getting equipment and loaning it and building it up over a period of time is the short answer to that.
I have a question. How many students do you usually have in your program?
The students, I usually have anywhere between 15 and 20 students. And it doesn't ever feel very large because I break them up into groups. And so I'm really working with four or so students at a time. So the number of students, if it's too small, I'm going to say is a problem, because sometimes things aren't working. The PCR doesn't work in one group, or something doesn't work, and the other groups are successful, and therefore they're able to share their experiment and keep everybody going. There are controls built into the investigation where there's DNA that is purified from Arabidopsis that's provided, and so that DNA, when it amplified, so it verifies that PCR is working because the DNA is there. And so there's some controls that are built in there as well. I know that doesn't directly relate to class size, but it made me think of that, because you don't, obviously, if you have like a large number of students, things can get problematic, but also small sizes are problematic too. But
I think biorad also has enough for 8 groups usually.
Yeah.
I am teaching a biotechnology program. We have 150 students.
Okay. Yeah.
And we're using case curriculum, which is kind of based on biorad kids. So
No,
but again, 150.
Right.
And I'm thinking about.
Well, when you say you're teaching like that number of students in a biotechnology, I got excited. I know that there's a challenge with like money and like getting like all those but you're halfway there. You've got the interest, you've got the students, like you're there. You just need,
it's a vocational pathway. They are getting certified as biotechnology technicians at the end of their high school careers.
No, it's awesome. And and this, I for several years, things happen. Like there was a student in one of my classes who was just like so excited about this, and she said, my dad's like totally excited. He wants, and I'm like, what does your dad do? Like he works at Gilead Science in the Bay Area biotech company, and I go, really? And so then next thing you know, he's donating equipment, he's donating cash, and I'm getting sponsored by, and things snowball. And as I segue into this, like when you choose a plant, you can choose it for a variety of reasons. Like you can choose something that's less characterized. In other words, like something that's really random or obscure, and the students might be able to publish a novel sequence, or pick something that's really common, and so that you can see and verify the results on the GenBank and make sure. The best option is maybe do one that's common, one that's obscure. Where my story that I'd like to share with you is that I've always personally loved the redwood trees in Northern California, and my school in California was very close to a most spectacular grove of ancient old growth redwoods that was just the most beautiful cathedral of trees you can possibly imagine, called Muir Woods. And we would go there on field trips, and over the years before I started doing this program, and the ranger would take us around, and I would talk to the students, and the ranger is dropping the sadness on the students and showing them, you know, how redwood trees have been cut for the last 100 years. And though these parks, state parks and national parks, preserve, and I'm putting this in quotes, and save the redwoods, they're not saved. They're not genuinely saved. And the students are like, what do you mean? Like, no one can come into the park and cut them down. And then they're like, well, like disease and things can come in. And so we started studying this, and I started working with some researchers from the University of San Francisco who were studying frog populations in Central America, and how when the numbers of species gets low, they start to interbreed, and there's like genetic defects, and there's disease that can come, and there's not a lot of genetic diversity within the population. And so it goes into something called an extinction vortex. And so keeping diversity is in life is one of the most important themes of life, diversity, whether it be ecosystem diversity, species diversity, but even within a species. This picture shows these little voles, but I was more interested in the genetic diversity of redwood trees. And so the students and I were like, we had this aha moment. We're like, let's go up to Muir Woods and study these redwood trees, and we're going to see how similar the redwood trees are one another by cloning and sequencing the GAPDH gene of redwood trees. And they, oh my God. And so several field trips and ranger permission and Save the Redwoods League donations. And the students, we were out there collecting needles and then taking the needles back into the lab and extracting them. And we found that the extraction protocols from Bio-Rad were good, but there were some obstacles that needed to be overcome, specifically to the fact that we chose the hardest, most stubborn plant that you can, like these needles. You can't crack these things with the simple micropestle. We were trying to use liquid nitrogen, but it's tricky with younger students, and so we were using dry ice. And when it comes to the lysis buffer, we started going into the literature, and I contacted my alma mater, the University of California, Davis, and I had some Davis professors working with me, and they were like, okay, you got to use this PVP, which is a chemical that sort of negates these polyphenols that the redwood trees have a lot of, that interfere with the DNA extraction. And so we were students, I go, you guys have to help me. So they're writing emails to different professors. They're getting different protocols. We're trying all different things. Then the ranger gets involved and says, you know what we want to know? We want to know why there's albino redwoods. And I go, hey, slow it down. There's a lot to consider here. They're like, we've always wanted to know why some of the trees are albino and some of them are not, coming off of the same tree. And so we were trying to use GAPDH to look at genetic diversity within the redwood population. And we ended up using chloroform in the extraction. We were using RNA ACEs, RNA nuclease A. We were using protease K, using MERCAP. We were using all different things. And so a long story short is, Bio-Rad got involved in this, and I was contacting them, and they were helpful. And the research and scientists there were helping us. So the students were collaborating not only with themselves, but with the professional community that surrounded the school. We were receiving grant money. We were allied with the Park Service. The parents were involved. The whole student body was involved. And Bio-Rad wrote a beautiful article. I put a link in the slideshow explaining the project, and it was miraculous. We were featured in a bulletin from the Save the Redwoods magazine. featuring our project. The students, I presented, but I didn't really. I brought my students in and we presented at a NSTA conference in San Francisco. These are all career moments for me. This cloning and sequencing, it's meaningful in a major way. The students were so into it, but it was funny is all those trips to the redwoods, then all the rest of the work was in the lab, pipetting small microliter amounts and things. So I thought, we got to get back out into the woods. And so the students wanted to do camping in the redwood forest. They wanted to do zip lining in the redwood forest. And so it became this redwood tree obsession that lasted over about 6 years when I was there. I'd like to think that I'm humble, but I'm really proud of this. I was on the cover of a Bio-Rad flyer in front of this redwood tree. That was a cool moment. And then it culminated in a biotech award at NSTA. And I can't tell you what this meant for the students when they won that award, not me. And so then other students wanted to get involved, and I'm like, well, we can do more, I guess, than just the redwood trees. Bio-Rad's got all these like cool kits. They have one on biofuels where you extract enzymes that break down cellulose from mushrooms in an attempt to produce a free glucose and then fermentation into ethanol, and then use all kinds of creative novel inquiry by selecting all different kinds of mushrooms to see what mushroom has the best enzymes in the reaction based on pH or temperature and things like this. And so I'm thinking, what we can do with this, we can go outside. And so I had some students, actually my daughter was one of them. We went out to a University of California reserve, and her and her friend were collecting exotic mushrooms, and we ended up writing real science papers for these experiments. And then you're like, well, how could they be published? We made our own journal. And so we were making our own journal of articles that we were doing, and we were conducting our own symposium at our school. And then Bio-Rad released a variation of their DNA barcoding where they use the cytochrome, the CO1 mitochondrial gene that can help determine the species of fish, if you're familiar with that fish DNA barcoding. But they also included now primers for mushroom and insects. And I was like, mushrooms? Insects. And so now the students are going out and identifying exotic types of mushroom using DNA barcoding. And then a student was interested in marine biology. And so I contacted a researcher at the University of California, Davis, where they have a marine station in Bodega Bay. And this is a professor at Davis was working with one of my students using DNA barcoding on mussels, because you're like, well, don't you know the mussels by just physical morphology? Not when they're juvenile. Even the researchers don't know. And that's important. I won't get too far into it because of the invasive species of mussels that's coming into the California coast. And the researchers need to use DNA barcoding to identify and in what areas are these mussels, distribution. And so the student was working with DNA barcoding for several months in Bodega Bay, and she published this paper, and she presented a symposium on it. She ended up going to Brown University and majoring in marine biology, and now she's working on her PhD. And I'm going to say it's because of Bio-Rad that happened and the kits. And so you might be familiar with sampling rivers and looking at macroinvertebrates, these little aquatic larvae that are on the bottom of rocks, and you try to identify it and try to ascertain the health of a river based on what species are there. and their abundance, and whether or not they're tolerant to pollution and various conditions. But now we are using DNA barcoding to identify the species in the river. And so this is a really cool investigation. Students are loving that. And then this got crazy when I moved to Vermont. The big topic in Vermont is certainly not redwood trees, it's maple trees here, but it's farming. And when I was talking to people, they were the public is so upset at genetically modified crops. They were like, people were like, Ray, you've got to teach these kids the evil of genetically modified. I'm not sure if I can do that, because I'm not convinced completely that is the case. The students need to come up to their own conclusions, as it turns out. So Bio-Rad's GMO kit looks at a PCR of food, and it looks at a couple of genes to see if they've been genetically modified or not. And so we did that in class, but then we took it out into the various farms surrounding my school in Vermont. And so the students began this project where they were just collecting leaf material from all different farms to see if it's been genetically modified. And I'm going to say it got a little controversial, because some of the farms were organic and they were offended by this, but they allowed it, and the students were good about it. And so we went in and we extracted the DNA, and we tried to look at different aspects. And so I introduced them to lateral flow strips, which is basically like ELISA. We also did an ELISA, looking for a protein that genetically modified plants express to fight off insects. You might be familiar with it. We used STS-PAGE to isolate the protein from that. And then we also used PCR to determine whether or not crops have been modified. And then we put all those experiments together in a journal. And we had a symposium at school, passed it out to the parents. They loved it. Several of the kids in the class are majoring in biological sciences. One's going to study biotech at Middlebury, and I believe he's there right now doing that. And this all culminated in going to workshops. I saw a movie called Evolution of Food, and a researcher I got in contact with from UC Davis, who then Pamela Ronald, who then zoomed into class. I got a researcher from Monsanto to provide the pros and cons of GMO. And then I got a professor at the University of Vermont became interested. He works in plant genetics, and we became friends. He was so impressed with the work that we were doing with GMOs, and he asked if any of the students wanted to visit his lab. They were welcome. So I had students over the summer interning at the University of Vermont. This is one of my students. And then I became good friends. I started working in his lab, and we were sharing ideas and equipment. And then all of that culminated in another NSTA award in agricultural science. And I think that was a pretty big deal, coming from a young guy who grew up in San Francisco. And so that's my presentation. And so thanks for your attention on this. And I also just wanted to finish by saying Bio-Rad has all of these resources. If you haven't been to the website, it's biorad.com classroom resources. This was just the tip of the stuff that they have there. There's guides, there's videos on YouTube, there's all kinds of case studies and information. It's a plethora of good. And so if you had any questions at any time, you can contact the curriculum and training specialist. You know, Tamika right over here, she's dominating the East Coast. That's where we are in Vermont. And so I can take any questions or comments regarding the presentation.
Well, thank you very much, but I do have a question. What is the age groups that you're working with again?
What groups? Well, I was working with
Age group, how old are your students? Are they freshmen, sophomores, juniors?
The students will are mostly juniors and seniors that I work with, but I in the question maybe, well, like, you know, how can they handle this? Well, if they have an introductory biology class, then that class seeds them in sort of the fundamentals of genetics and et cetera, and then it enables them to be able to branch out in their junior and senior year to do these independent research classes. So juniors and seniors are the ones. But I have to tell you, the research class becomes so popular that younger students want to get into it, and it's hard to say no to them. But it's a bit much to take on, because it's not only the information, but just like everything I was saying, like connection to researchers and things like that. And so the older students are a little bit better. And this certainly works out at a college level. I had a student who graduated who went to USC and then reached out to me and said, guess what? In one of my biology classes at USC, we're doing the cloning and sequencing series just like we did in high school. And I said, well, pay attention. You might there might be more stuff to be to learn. Maybe you can help others in your class as well. If you know, it's one of those things. So I'd say high school and college level for this particular cloning and sequencing.
Okay, thank you. Yes, definitely. I agree with you, Lizzie.
Yeah.
Well, I'm working with freshmen.
Yeah. And so